Good morning, everybody, and welcome to the second day of our single-cell proteomics conference. I had a wonderful time yesterday, a lot of exciting talks, and I'm very, very much looking forward to the talks of today. And just at the beginning, I want to tell you a little bit about the end of the day, when we are going to get together and discuss some topics relevant to the entire community so that perhaps we can arrive at some useful consensus conclusions. And here I have provided a few suggested topics that we might want to discuss, such as what are the best benchmarks for single-cell mass spectrometry, how can we share data and software in a way that will be most useful for everybody? What are the biggest marginal gains? We all have our favorite things to focus on, which is truly wonderful because it is this diversity that can contribute to the growth of the field, and I think that can make for a fun discussion to see what various people think and what are the pros and cons. And of course, I'm looking forward for more suggestions. So each topic is going to have its group leader, and they've already nominated some group leaders. For example, for data sharing and software integration, I think that Jorgen Cox would be the best group leader that I can think of. Using MaxQuant was one of the common threads in many of the talks yesterday, and I know that Jorgen is already thinking of ways how he can integrate various software that different members of the community are developing. So, if you're interested in leading a group discussion, let me know. We are going to have breakout sessions of about 30 minutes. We are going to discuss these topics within each group, and then we'll come together, and the group leader of each group is going to present a short summary of the discussions. And on the topic of data sharing, since there was a lot of interest yesterday in the data that I presented, I went ahead and uploaded the raw data online. So you can access this from the conference schedule. If you just click on my presentation's title, you can get this link, or just from this link you can follow and you can download the raw data, and there will be a lot more coming soon. All right, and now we are moving to something very, very cool. Many of us here are aspiring pioneers, but some are successful pioneers. And we are about to hear from one such successful pioneer who, 25 years ago, laid the foundations for so much of what we do today, how we analyze mass spectra. And I cannot imagine modern mass spectrometry without many of the ideas that John Yates has pioneered. So I'm very, very much looking forward to hearing about the early days of Sequest, how it was born, how was it invented.
Okay, thanks. Thanks, Nikolai, for the invitation to talk. I don't know how you got it in your head that I should give this talk. This is something I put together a few years ago for the 20th anniversary of Sequest publication for the US HUPO meeting. So it was in Seattle, so it was kind of fitting. A little bit of a pretentious title, but good morning. I think it's interesting to look back because I mean, what's interesting is to look at what the surrounding technology was like at the time. I mean, we have more power in our cell phones now than the computers did back then, and laptops really weren't invented. The internet really wasn't invented. Lots of different things that made it kind of hard to think about doing these sort of strategies. This is it's a favorite quote. My wife says I shouldn't quote movies, but I like to anyway. This is really kind of illustrates the shifting landscape of knowledge. We're always very convinced of something until we're not. We're convinced that the world was flat until we weren't. And what often drives new knowledge is that we have technologies that allow us to ask these questions. Like you guys are driving single cell analysis, and that's going to open up and change our perception of a lot of things about biology. And I mean, that's not a new concept. Freeman Dyson said this quite a while ago, 1997. New directions in science are launched by new tools much more often than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have to be explained. I mean, just think the vast knowledge that we now have because of genome sequencing. Lots of things that we didn't understand before that we now have a much better grasp on. Although, you know, what's been interesting to watch in the genomics field is, you know, you sequence one genome or sequence one disease genome, and well, the answer didn't fall out, so we got to sequence 10 more genomes, and then 100 more, and then it's 1,000 more, and then it's 1,000,000 more. So I think some aspects of genetics are going to be difficult to explain, and just on genetics alone, and so it's probably up to the proteomics people to try to figure out what those diseases really are based on. Okay, so in 1989, with the Human Genome Project looming, the future of protein biochemistry was uncertain. This had appeared in 1978 in Nature, no less, the decline and fall of protein chemistry, and what happened that precipitated this? Anybody? You guys are all too young to remember. 1977, publication of Sanger's paper, Rowley Gilbert's paper on DNA sequencing, and all of a sudden, all proteins were going to be sequenced by their genes. And so this guy wrote this unfortunate article, how protein chemistry was going to go away. And, you know, if you think about it, he kind of is right, because in the end, all proteins were sequenced by DNA sequencing, and we created a database, and we're just adding the decorations that go onto them and some of the fine details. Also, 1989, around that time, the National Academy of Sciences met and wrote a report on whether or not we should actually sequence the human genome. It probably was kind of a done deal at that point, but this was the argument to Congress to put up the $3 billion to sequence the human genome. And nowhere in that report does anybody say, if we sequence the human genome, that's really going to benefit protein biochemistry. It's all about bioinformatics and diseases and so on and so forth. And new drugs would come of this. And on the 10th anniversary of the human genome being sequenced, there were a bunch of scorching articles that came out in newspapers about, okay, where are our new drugs? And I guess it's not that easy. And I finished up with my PhD, and I stayed on for an extra year at Virginia because the FTMS was coming online. I wanted to be around for that. And then I went to Caltech, to Leroy Hood's lab. Is there a pointer up here? I guess if you don't know what Lee looks like, that's Lee right there. And this isn't Lee's group. This is just a biotechnology course that we were teaching, and scattered around are a few people. If you can find me, then good. This is Debbie Nickerson right here. She went up to the University of Washington when Lee moved, and she's been running her own lab. And one of the huge differences between proteomics and genomics during the ARA funding, remember the supplements that came out in 2009, I guess, she got like $50 million to do DNA sequencing, contract sequencing for NHLBI. Bought a bunch of Illuminas. Bought so many Illuminas, they put her on their scientific advisory board. Tim Hunkapiller is back there. Tim is the brother of Mike Hunkapiller, who's applied biosystems fame, also the person who was instrumental in building the gas phase sequencer. So at this time in Lee's lab, all the talk was about human genome this, human genome that. Lee did have a significant protein biochemistry group, but I mean, the big names in genome sequencing were marching through Lee's lab, coming to talk about, you know, the strategy and to see what Lee was doing, because he had the new 4-color DNA sequencer in the lab and was using it, and they were trying to figure out, do we do directed sequencing, do we do shotgun sequencing, how are we going to sequence the genome, and so forth. So it was an interesting time, but a little bit depressing for protein chemistry. And so what was going through my head in that time was, what's going to be the role of mass spectrometry and protein biochemistry in the era of genomics? And When I was in grad school, that's Don Hunt, I did my PhD with, the fight had been with the Edmund sequenator, which is this thing. And this is what was invented in Lee's lab, the gas phase Edmund sequencer. Anybody here know Edmund sequencing? Yeah, so a few people. And also the big thing was 2D gel electrophoresis. You know, Rudy Abersol's claim to fame nowadays is swath and all this stuff, but back in the early 1980s, and he was at Caltech as well. Actually, I missed him by about 2 weeks. He left 2 weeks before I showed up. His thing was pulling spots off of 2D gels and then sequencing them. And one of the things that we were thinking about at Virginia when I was there was pulling spots off 2D gels and then sequencing them on the mass spectrometer. But anyway, this was the deal at the time. And so it wasn't clear what role we were going to play in the future of genomics. And anybody recognize this? It's the Hollywood sign. The undergraduates at Caltech are extremely clever, and so they figured out how to drape the Hollywood sign with sheets to spell out Caltech on the 100th anniversary of the founding of Caltech. They're renowned for their pranks. One year they, during the Rose Bowl, they got into the scoreboard and they changed the score. It was MIT 0, Caltech 99, something like that. So they were pretty clever people. All right, so this also appeared, and in science, protein chemists gained a new analytical tool. Anybody know what it was? 89, electrospray ionization. I think this issue may have been the issue where Fenn had a paper on electrospray ionization. But discussing about mass specs and so forth, and this is a figure out of a paper from Klaus Biemann. It really is kind of illustrating a 4-sector mass spectrometer. And so this goes through discussions about that. I'll point out just a few things. So Don Hunt was quoted as saying, his vision of the future, protein chemistry labs will have 2 mass spectrometers for every sequenator, where now you're lucky if you find a mass spectrometer for every 6 or 7. But his critics argue that no one but Hunt has been able to extract sequence data from a quadrupole. So the other big fight going on at the time was between people with magnetic sectors and people with triple quadrupoles. And Hunt was the only advocate of the triple quadrupole at the time. And in fact, Steve Carr gets quoted. Others are waiting for electrospray or laser desorption to be adapted to magnetic sector machines, which have greater resolution and a more proven record in protein sequencing. When that happens, the sky is the limit, predicts Steve Carr, a mass spectrometrist with SmithKline and French laboratories. So before Steve came back to Boston, he was at SmithKline. Anybody ever see a magnetic sector? Any recently? So magnetic sectors were the Orbitrap of the day. So high resolution, high mass accuracy instruments, they were the workhorse of organic chemistry. And the problem was they were very slow to scan, so you couldn't do electrospray on them easily. You couldn't adapt the electrospray source onto a magnetic sector because the source in a magnetic sector instrument floats at 10,000 volts. And then they scanned so slowly that you just couldn't do LC. And so it was like the asteroid hitting the planet and the dinosaurs disappearing. Magnetic sectors were gone. They're now still used a little bit in environmental analysis. They're really good for doing dioxin analysis. Okay, mass spectrometry was potentially very fast. You could increase your scale of measurements, increase sensitivity of measurements, generate new types of data, increase throughput, more experiments as a function of time. But in the field of mass spectrometry, back in that time, the bottleneck was data analysis because you had to do everything by hand. Computers had a very early role in mass spectrometry, so Beeman's lab was a leader in this area and developed methods to acquire, display, store MS data. So really the goals back then were more aligned with just trying to instead of putting down a mass spectrum on heat-sensitive paper or a photographic plate, to try to get it into a computer so that you could use that data in some fashion. They also developed data analysis methods, including library searching and some peptide sequencing algorithms. Those came very much later. But Galafri's lab was very involved in library searching. Joshua Lederberg, who got a Nobel Prize at a very young age for some bacterial thing, bacteria having sex, I guess. He was, when he was at Stanford, got interested in AI, and they picked mass spectrometry data interpretation as an intellectual problem for some of the early AI algorithm development. They developed this program called Dendral, which was supposed to help interpret a mass spectrum like a human would do it. And it actually did pretty well, but computers were probably not powerful enough back then. So in Beeman's lab, they had one of the more powerful mass spectrometers of the day, that IBM 1800. I don't think this is the original one, but it was of this model. And so this is what they were using to do a lot of their data acquisition and some of the data interpretation and so forth. And yeah, this is probably not even as powerful as that flip phone used to have. All right, so computers and mass spectrometry. Main limitation of mass spectral data analysis for peptides is interpretation. De novo analysis was largely limited to a spectrum and was not particularly accurate or general. Data quality is always the main issue. There were a few attempts to make computer algorithms to do this. Marshall Siegel, back in '88, had a paper, and then Klaus Beeman's lab, Richard Johnson, who's still active in mass spectrometry, also had a paper to interpret high-energy collision tandem mass spectra. So that's a peptide spectra that came off of a four-sector mass spectrometer. But this is mostly how mass spectra were interpreted. You'll note that there's a PC here. I was in graduate school, and my wife kindly let me buy a computer. It's an AT&T PC 6300, 8 megahertz, 8086 Intel chip, dual floppies, 64K of RAM. So this was a smoking computer at the time. I had to have the dual floppies. And then this is the literature right here on how to actually operate the stupid thing. But you'll note that I'm sitting there trying to interpret tandem mass spectra. So I was motivated to find a better way to do this. So before there was NCBI, which was founded in 1988, GenBake was managed through a contract to IntelliGenetics. And they had a newsletter. They were a company of some kind, and you can see to access GenBank here you had to call your local telnet node. They're going to have a users meeting in Bethesda. Let's see what else. So it wasn't straightforward to access whatever data was in GenBank at the time. This was 1988. The other thing that this company did is that they sold a program called PCGene. And it was programs that had been developed by Amos Baroque and the others at Swiss Prot. And they sold this. We bought a copy, had a bunch of programs. But what it also had was a database of sequences, which got me interested in sequence databases. The problem is that it was in binary, and I kept trying to print it out, figure out how to print it out, and I couldn't get out the flat files. Of course, because it was in binary, because it was on a floppy disk, so it had to fit. But one of the things that we were doing with this was sort of a predecessor to peptide mass mapping. And I had written this up as an applications note for this particular newsletter, where we would sequence a peptide and then use that sequence to do a FTP scan or a scan sim through PCGene, and that would pull back some protein sequences, and then we'd take the masses of peptides, tryptic peptides that we'd found, and we'd try to match them up to the sequences as a way to avoid trying to sequence something that's already known. Because almost everything we were doing back then was de novo sequencing, or trying to do. All right, so I also was involved in trying to write programs to do de novo interpretation. This is a book chapter on one of the programs that we had written. Wasn't all that successful. Okay, so at this point, I've now gone to University of Washington. I've started my own lab, and I have a tandem triple quadrupole mass spectrometer. And people know triple quad. So triple quads were used primarily to were the sort of workhorses at the time. There were no such thing as ion traps. FTMS was around, sort of. So this is what we were using to do protein sequencing mass spectrometry. And so in the lab, we were sequencing class 2 MHC peptides in collaboration with an immunology lab We would partially sequence about 4 to 6 residues, then we would blast search the partial sequence by emailing the sequences to the blast server. We would use the hits to make sure we weren't spending time on no known sequences. And so while sitting there waiting for an email to come back for the blast search, I started wondering why not just search the spectra directly against the sequence databases. That was sort of my aha moment. So there were 3 problems to get to a solution for this. So the first was data access. So we had to access MS data. That's like now, it's in a binary format, proprietary format. And it's actually a federal regulation that the data has to be in a proprietary format. You can't access it. You certainly can't take data out, modify it, and put data back into the file. That's a big no-no as far as the FDA is concerned. And then how to get sequence data. And to give you some perspective, so this is like 1992 or 3. To give you some perspective, Mosaic Netscape 0.9 was launched October 13th, 1994. And so the World Wide Web really didn't quite exist until this program came out. And then how to compare spectra, then how to reconstruct spectra from sequence. So if we have sequence, we get a tryptic peptide, how do we match that back to the spectrum? You know, all those sort of issues that we have to think about. So we had limited access to data in 1992. NetCDF was a standard, but it looked to me like a manufacturer's slow walk to appease customers. That the manufacturers really did not want to give you access to the data. And they gave you this program called NetCDF. I had talked to Ian Jardine, and he gave me, Ian Jardine at Thermo at Finnegan at the time, and he gave me a program to output an MSMS spectrum, single spectrum, into a flat file format. So it's very difficult to do an entire LC-MSMS run, but at the time, DDA didn't exist. So we sat at the computer and we basically typed in the masses of peptides that we wanted to get sequence information on. And I got Finnegan's attention when Jimmy Ying, who was a programmer in my lab, cracked the Finnegan DAT binary file format. They immediately sent me an NDA, and they gave me access to libraries to get the whole spectra out so we could convert all the data. So that was one way to get their attention. So, you know, now getting access to all this data is really simple. We could download databases like GenPep. GenPep was the protein sequence database in 1993 by using FTP. And so we could get access to that database. It might take a while to actually get it, but we could get it. Okay, so the plan was to pre-process tandem mass spectra, eliminate noise while not losing weak signals, convert the nominal M over Z, remove precursor ion. This is TSQ data, so there's a very large precursor ion still left in the spectrum, and it could dominate any of the mathematics that you'd use to try to compare spectra. So we kept 200 most abundant ions. We noted the ammonium ions during pre-processing. So now that we've got, when we were mostly ion traps, we lose all the low mass M over Z fragment ions, the ammonium ions, which tell you something about what amino acids might be present in the spectrum. A few of the amino acids anyway. But in the triple quad spectrum, we still had that data. Now with HCD, you have that back, so you can use that information. So you can tell if there's a histidine, a methionine, a tryptophan, a tyrosine, or a phenylalanine almost immediately in the spectrum just based on the ammonium ions being present. So step 2 was to search, calculate molecular weight of peptides in database. We did it with no reference to proteolytic cleavage because we were influenced by the fact that we were looking at MHC class 1 and class 2 peptides. And so we wanted to preserve our ability to do that. And we've just continued to do that to this day. Preliminary scoring, so we came up with a way to try to do a preliminary scoring and ranking of scores based on this formula. If you want to know what all that means, you can go back to the original paper. Oh, it's right here, actually. Ion intensity of observed fragment ions, N equal number of predicted fragment ions, beta is fragment ion continuity. So if we had a continuous series of fragment ions, we gave it an increase in score. Ammonium ions were present, and that was present in the sequence. We gave it a boost in score, and so forth. And then the biggest thing was how to do a spectral reconstruction and cross-correlation analysis. So there were lots of things that we could learn from library searching, but one of the things is that we had a sequence, and we had to do something to try to match the sequence to the spectrum. I had a thing in my head on how I wanted to do that. So So we did noise reduction by removing all but the 200 most abundant ions. Then we divided them to 10 equal sections, and ion abundances were normalized. This is what a typical mass spectrum would look like. And so one of the reasons that we did this was to try to boost up low-level signals in certain sections of the spectrum. And it looks very noisy, and triple quadruple spectra back then were, in fact, pretty noisy. But what this was intended to do was sometimes the fragment ions in this end of the spectrum would be weak, and they'd be dominated by fragment ions that are in the lower end of the spectrum. So we wanted to try to eliminate that kind of bias. And then this is how we reconstructed an MS-MS spectrum from sequence. So we threw in water losses, ammonium losses, and then the B and the Y ions, and we gave them all a certain height of 50% intensity. So rather than try to predict fragment ion intensities based on amino acid sequence or something like that, we just did this as a way to normalize it. Then we needed a way to do the comparison. And for some reason, I got in my head that we should do a correlation analysis. And there was a paper that we found by Kevin Owens at Drexel University where he applied correlation analysis as techniques to mass spectral data. So he's basically using cross-correlation analysis to compare library spectra. And that came from the fact that in the Heifja lab back in 1978, they used cross-correlation analysis to compare infrared spectra by correlation-based file searching. Kevin Owen was a graduate student in the Heifja lab, and so in the Heifja lab, he also applied cross-correlation analysis to mass spectra while he was there. And so that looked like a pretty good way of doing this. And so this is what we came up with. So we would pre-process sequences out of the database. It's like this. We'd pre-process the tandem mass spectra like that. And then we'd do a cross-correlation analysis using fast Fourier transforms, inverse FFT, and then we came up with a way of evaluating this, where we took score out from 75, range plus 75, minus 75, and so on. And then we came up with a cross-correlation score for how well these two things fit together. And once we got all this put together and we ran our first search through a sequence database, and mind you, this was on a DEC workstation. It took 2.5 hours, one spectrum. So we had to rethink the process. It was a little bit slow. But eventually we got to the point where we could do searches in a reasonable time frame. This was from the original Sequest paper, where we took a Saccharomyces cerevisiae lysite and we digested it up with trypsin, and we sequenced. And again, we're typing in the numbers as for peptides, and we sequenced a bunch of peptides. Showed that we could sequence a bunch of peptides using this algorithm. So this was sort of the start of shotgun sequencing, but we didn't have DDA back then. And then this is another subsequent paper where we did the same thing with E. coli lysate. And again, we're putting the numbers by hand to sequence peptides. And so we're not getting very large numbers out of these things. So this finally got published. So the story here is send it to Fred McLafferty. Fred McLafferty is famous for library searching and mass spectrometry, member of the National Academy of Sciences. And he spent a large portion of his life working on library searching, among other things. Roman was a scientist there, right? So postdoc? Postdoc. So I thought, he'll understand. So I sent it to him, and he sent it out for review, got the reviews back, and rejected it. I think he reviewed it, and I know Klaus Beeman reviewed it. And Fred gave me some bad advice. He said, submit it to another journal, and then have the editor contact me, and I'll give him the names of the reviewers, and that'll speed up the process. So I did that, and sent it to Protein Science, and they must have contacted Fred, and he gave him the names. And so I don't think Fred reviewed it again, but I think Beeman, I know, reviewed it again, because he told somebody that he did, and got rejected again. And the editor, Russell Doolittle, took pity on me and said, oh, revise it and send it back. So I revised it, sent it back, and sent it back out to Beeman and whoever else, and they rejected it again. So I got 3 rejections. And so the only reason this got published in JASMIS is I was bemoaning my fate to Don Hunt at ASMS, and Don says, you got a copy of the paper? I said, yeah. He said, give it to me. So I gave it to him, and he marched it over to Mike Gross, who was the editor at Jasmus, and handed it to him and said, Mike, you need to publish this. And he read it. He said, this isn't so bad. And then he said, respond to the criticisms, and yeah, I'll publish it. So I did that, and then he published it. Okay, it wasn't just getting these things published that was a problem. This is another quote from some movie, I think. Second mouse gets the cheese. Trying to get grants funded was a problem. And this was actually my second attempt, and I get you can see it up there, percentile rank, 74.3. This was under the old scoring system of 100 to 500. So that's about halfway in between. And there's the major comment is the reviewer is telling me I should write an algorithm to sequence the peptides and take the sequence and then use FASTA to search the database. And who's on the committee? But William Pearson. And what did William Pearson invent? FASTA. So I never actually had any direct funding to work on this program. And then that's the letter from Russ Doolittle rejecting the paper. But we got it published, and then very quickly we worked on trying to do the same search with modified peptides. This got published, this one a lot smoother, it went into analytical chemistry. One of and again, it wasn't particularly efficient, although efficient way to do these searches, but it was effective way to do these searches. That did in fact work. And so this is for phosphorylation analysis, where we consider within peptides every single possibility of how it could be phosphorylated within the parameter of the molecular weight of the peptide. And this is a DDA experiment here. So the other lesson I learned is not to listen to people too carefully, because I asked engineers at Finnegan, and the instruments back then had this thing called instrument control language, where you could write programs to direct the mass spectrometer to do things. I mean, all that's done for you now. But back then, we used to be able to do it ourselves, and it was fantastic because you could innovate. And so I asked one of the engineers at Finnegan, could I write a program to automatically collect tandem mass spectra? He said, no, that won't work. And so I ignored, so I didn't do it. And then Terry Lee, who was at the City of Hope, did it. And so he published it in 95, and so this also came out in 95. And so we were right behind him. But I didn't do the exact same thing he did. What I did is I wrote a program to automatically collect phospholipeptides by looking for the neutral loss of 49 or 33. And then we used this. This is one of the first papers we published where we used the algorithm to look for phospholipeptides. So with Mike Gelb. And here's one of the phospholipeptides, beautiful spectrum. There's another one, almost a beautiful spectrum. You can see it, the spectra back then were a little noisy. Okay, so the next thing we did, this is also again published in 95, is mining genomes. We wanted to do is to also use nucleotide databases, so the cDNA databases, which were starting to emerge, to be able to search them directly. And so we do a 6-frame translation and try to match up tandem mass spectra to the scores. And so we would do a 6-frame translation and we'd match them up. And I was a little bit surprised that given because the numbers of sequences you were considering went up pretty dramatically. And so I was pretty surprised that we were still able to get pretty good matches to things using this sort of strategy. And we used this off and on to do what's now called proteogenomics. And so for this particular project, the Plasmodium yoeli, we did all stop-to-stop ORFs greater than 100, and we searched to help identify the genes in this particular genome project. And some famous people on there. See, Craig Ventnor, Claire Fraser. All right, the other thing that we did that was kind of interesting was to look for amino acid sequence variations. And again, this was not a particularly efficient way to do it, but one of the things that Tina Gatlin did is, Tina's now a program officer at the Genome Institute at NIH. She developed a strategy which we now call triple digestion, where we use trypsin, elastase, subtolysine as a way to get almost 100% sequence coverage on proteins, and it works really well. And the problem with elastase and subtolysine is that they're non-specific enzymes, and so they create peptides that make people doing database searching nervous, because you have to do a non-preference type of search, which is what we were used to doing, so we weren't the least bit concerned about it. And it worked pretty well. We developed a version of Sequest which we called Sequest SNP, and we would look at all the potential variations, but we do it in a gene. And it was reasonable if you weren't trying to search an entire nucleotide database. But we then used this to identify some variations in a hemoglobin gene used with the Laboratory of Medicine at the University of Washington. Okay, so by '96 we have the ability to identify peptides, protein modifications, and assist in gene annotation. We also published a library searching paper in 1998. This is now much more widespread. And in this period of time, people were not sure you could do library searching with tandem mass spectra because there was a fair amount of variability in the spectra. And so one of the things that Fred and others had done for library searching was standardization. So the 70 eV electron impact spectrum was the standard of the field so that you could enable library searching. And when tandem mass spectrometry came along, it created this variability that people weren't comfortable with. And so people didn't seem to have moved into this idea of doing library searching based on tandem mass spectra. And so we published this paper in '98. And one of the goals of it was to show that we could match ion trap spectra to triple quad spectra and vice versa. And our libraries weren't that big, a few thousand spectra or something like that, but we showed that we had a method where we could actually do that comparison. Okay, so what we need now so the yeast genome sequenced in '97, '95 Haemophilus influenza. What we need are completed genomes, they're coming. Fast scanning mass spectrometers, the LCQ had just come out, and that was way faster than the triple quad. With a triple quad, we were collecting one tandem mass spectrum every 3 to 4 seconds. On the LCQ, we were collecting 3 or 4 tandem mass spectra every second, something like that. So that was a big improvement. Faster computers. One of the things that we did in the mid-'90s was we built a Beowulf cluster, which was fun. Rather than buy an actual computer with 4 processors in it, we just built a Beowulf cluster because it was much cheaper and also much more fun to do that. Data acquisition algorithms and data transfer, large-scale chromatographic separations. So we started dabbling in LC-LC, and then we have to do biochemical methods suited for mass spectrometry. So lots of things to do. So, we don't really appreciate it right now, but this idea of automation on the instruments was not a thing until about 1990 when Finnegan invented this idea of instrument control language. So putting a embedded computer on the instrument that would run the operation of the instrument. And these guys, Sokolov, Steiner, and Lewis, invented instrument control language at Finnegan, which is the basis for everything we do now do you think about if you had to sit there and type in numbers? And so that enabled data-dependent data acquisition and the ability to look at a peak and then decide whether or not you want to do tandem mass spectrum on that. And that was done first by Terry Lee's lab. Came out in '94 instead of '95. We followed this very quickly in '95 with our own algorithm for doing it, like this, which I already showed you. And we used that to look for phosphopeptides. The other thing that was important too is, and this was these were developments that were going on almost in parallel to what was going on in the mass spec field, is the chromatography people were experimenting with nanoscale columns or microscale columns at nanoscale flow rates. And so this dovetailed really nicely into what we needed in mass spectrometry to scale down the flow and to improve detection limits. And I mean, Jorgensen certainly, they were trying to investigate wall effects in chromatography using these strategies rather than thinking, oh, those guys over there are doing mass spectrometry, they're going to need these small columns someday. So it just worked out beautifully. People have been thinking about two-dimensional chromatography for a long time. I always thought this was the coolest idea for doing separations. And it was certainly important back when mass spectrometers didn't scan very fast to be able to spread things out. And Jim Jorgensen's lab was also developing two-dimensional methods to do separations. He wasn't the only group, but he certainly had a major influence on me. And so they had, by 1990, they had developed strategies for doing two-dimensional separations. I didn't like them. I didn't think they were would be very effective for mass spectrometry, but they certainly gave me inspiration to try to develop other things. And we bought this box for doing two-dimensional, multi-dimensional chromatography. This is from a company that's no longer in business, Perceptive Biosystems. It's a device called the Integral, and we used it in a 1999 paper to separate yeast, and we got 189 proteins. Doesn't sound like a lot, but that was the record at the time. The previous record was 160 by the Mann Lab off a 2D gel, which I'm sure took a lot longer to do than this. This was 24 hours. But this used large-scale columns. These were 300 micron columns or something like that. So they were actually quite big. And so we developed what became known as mud pit in a 100 micron fused silica column as a way to scale down separations and flow rates and try to improve analysis. And then when we did that, we increased by almost an order of magnitude the number of proteins that we could identify, which again was another record done in about 24 hours. And again, this was done on the same mass spectrometry technology as that previous study. So we got an order of magnitude increase in proteins just by scaling down the chromatography. And then that brought us to, this point where we started doing more and more shotgun proteomics, digested the protein mixture, the peptides, and then the automation at the mass spectrometry end and the automation at the interpretation end allows us to do much more large-scale proteomics and get at all these sort of questions unanswered by whole genome sequencing, post-translational modification, regulation of function, covalent modification, protein localization, protein-protein interactions. The Gigi's lab is beating this one dead by doing 10,000 pull-downs, the protein-DNA interactions. So it was this convergence of technologies in this time frame of 89 to 94, which gave us proteomics. And this was, I think, one of the great unintended consequences of genomics. And I've talked to a number of people that work in the genomics field, and they actually think that proteomics was a huge benefit from the genome projects, that microarrays and things like that, but that proteomics certainly was one of these great unintended consequences. So the paper did pretty well in JASMUS. So it's almost at 6,000 citations now. The yeast paper, doing shotgun proteomics, 5,000 citations. This is this paper in JASMUS. I think it's the most highly cited paper in JASMUS by maybe a factor of 6 or 7 now. So Mike Gross was pretty happy. So more importantly, the technologies and innovations enabled high throughput and large scale analysis, was this sort of thing, and that just keeps going on and on. And so this was my group at the time. This kid that looks like a high school intern, that's Jimmy Ng. He was actually 3 years out of a master's degree in electrical engineering from the University of Washington. So he wasn't just hadn't just finished undergrad. He was a graduate student at the University of Washington. And on his CV, he had that he'd worked with, as part of his master's program, he'd worked with neural nets. And that's what caught my attention, and then I hired him to work on this project. And let's see. So that's Andy Link, Dave Schultz. Andy did the first yeast paper, and Washburn's not in this picture. So Andy and Dave worked very closely together on the first project. And that's not quite my current group, but some members are still there. And so thanks.
Thank you, John. I certainly learned things that I thought are relatively new, have been going on for a long time. For example, the 6-frame translation that now has become quite common, trying to identify novel protein products. I did not realize has such a long history. So thank you for teaching me the proper history of some things. Are there questions for John? Yeah. No.
Well, that's very interesting. You didn't cover this aspect when there was a competition with other approaches. And particularly, there was this tag approach, sequence tag approach, favored by Matthias and Matthias Mann. So, and for a while, there were like 2 competing approaches, and then your approach won because the tag has a big deficiency. It's only 70% reliable. And so your approach is completely blind, and therefore it always works. So could you comment on that, this part of the story?
So what Roman is referring to is about the same time there was a paper from the Mann lab. And if you look at the Mann paper, he cites my paper. I'd given him a copy of it at the Methods in Protein Sequence Analysis meeting. But they used a sequence tag. And so the differences in the process were he was doing nanospray infusion into a API-3 triple quad mass spectrometer. And unbeknownst to me at the time, the API-3 mass spectrometer could not do LC on the fly. They could not do MSMS on the fly. The collision cell in that particular device was very inefficient. And so the problem that Matthias solved was by nanospraying infusion into the mass spectrometer, they could signal average for a longer period of time. And so they could actually get very good spectra, but it took a while to get that, 5 to 10 minutes. And from that spectrum, they'd sit there and they'd watch it being collected, and they would start sequencing off the spectrum. And they would use that sequence tag in a search of the database. And then there were little tricks you could do where you would leave off the N-terminal mass and leave off the C-terminal mass, and so you could pick up things that might be modified. And so that was a good approach. It was an accurate approach. The problem is that it wasn't an automated approach, and it also wasn't an approach that lent itself well to LC MSMS. So it kind of died off for a while, but it came back when people started to automate the process of generating a sequence tag. It's much easier to generate a sequence tag than it is to sequence an entire peptide, because there are frequently missing fragment ions in the spectrum, things like that. And in fact, we did one of the automated versions of it, GutenTag, and Dave Tabb did that. Dave Tabb also did that later on when he started his own lab, and I think Pavel also did a version of it. So it's come back, and it's actually a reasonably accurate way to do things. But the original implementation was not an automated approach.
So when a lot of these things were happening, I was in my early teens. I don't have any...
in high school?
Yeah. Well, maybe even junior high. But
so next year's 25 years publication of that paper. So that's a quarter of a century. That's a long time ago.
Anyway, so my point was that I don't have any first-hand experience observing the community, but I've heard from others that there was a lot of hesitation for many colleagues to adopt the possibility, to accept the possibility that a computer identification can be as accurate as a manually identified spectrum. Can you tell us a little bit more about that? What helped
I think to this
convince them?
day, Don Hunt doesn't believe what comes out of the computer. But Don is he's amazing at interpreting these things by hand, and so he still works on these. For 30 years he's been working on cancer immunogens, so neoantigens, what's now called the neoantigen field, and he analyzes all the spectra by hand. He's always got a pile of spectra in his briefcase that he's interpreting. But yeah, there was a lot of junk published in the early days, and so there was some suspicion. Some people were just over-relying on what was coming out of the computer rather than following them up, and people were just publishing lists, and so there was a reason to be a bit skeptical of what was coming out of the computers at the time. But the big competition was 2D gels versus LC. I used to get invited to 2D gel meetings so they could make fun of me. Why aren't you doing 2D gels? Look at this; these are great.
John, also, you mentioned that it was very hard to publish the first paper. I know of another database search paper that of Peter Obster that was delayed by a year and a half and was ended up lagging behind the
Was that
curve.
the peptide mass mapping?
Yeah.
Yeah.
Well, something like that. So, would you comment on whether is this a feature or it's a bug? Is this a feature of our scientific community that we're very resistant to accept novel ideas, and why, and what eventually wins over the public opinion?
Yeah, some reviewers feel like you're trying to well, it's like you're trying to create a criminal act, and they're defending the integrity of society. I don't know. Some people are just, sometimes it can be a bit unbelievable what people are proposing or doing. But I think science is self-cleansing, and so I would err on the side of taking the risk that something will, if you're not going to follow it up, then it's going to disappear. In fact, much to my chagrin, Steve Stein pointed out at an ASMS meeting, I think it was 10 years after we had published the library searching paper, said, it was a very nice paper published in 1998 or 2000 or something on library searching, but there was never a follow-up paper. So, that kind of bites sometimes, but I could never get the lab to put together libraries. It was just too easy to do what they were doing.
Yeah, thank you very much for your talk. I really enjoyed kind of the history of how it did happen, because again, I was too young to kind of understand how it actually born. First of all, I feel much more comfortable to be and honored to be in the company of the people who has a paper that had been rejected for more than 1 year. I like it. I think it's a great actually company to be in. My question is right now, since this all started beginning, clearly computers improved, technology improved. We have amazing mass specs right now, beautiful C, at the current state, currently at 90-minute gradient, we have more than 1 million mass spectra could be acquired, right? According to all these things that improved, my general question that probably you're one of the few people I would like to ask this question. How you'd explain, with all those improvements and technologies and all this stuff and databases were absolutely complete, you never get a full 100% explanation of all those MS that comes in clearly peptidic nature. You see the peptides, amino acids. What are those? What is your opinion? What are those spectra that doesn't fit anything? We blast them, we try to apply amino acids. swaps with possible PTMs, anything, and they still not explainable. What that field is, what this cloud of the unknown MSMS, in your opinion?
Yeah, so Gidgey had a nice paper in Nature Biotech a few years back where they opened up the search window and they went looking to see what are those other spectra. And a lot of them are sequence variations, modifications that people haven't searched for, things of that nature. But I've always maintained that, you know, we do these runs, we think that everything in the run is a peptide, and that's probably not a good assumption. So there are background chemicals, there are probably metabolites in there, detergents, things like that. So not everything is going to be a peptide. And I would point out, I had this one, when I was a grad student, I had this one spectrum that I could not interpret. I could not figure out what it was. Well, I eventually did, which is how I know this. And it turned out it was a peptide with an oxidized methionine, and in the fast atom bombardment tandem mass spectrometry process, the sulfur fell off, and it created an unusual amino acid. And I just couldn't figure it out because I didn't know that the original peptide was, and what I did is I collected the MSMS on the peptide that had beta eliminated the sulfur oxide. And so I couldn't interpret it because I didn't know what that thing was. So it took a long time for me to figure it out. And there's probably a lot of that going on. Oxidations that we don't anticipate, a lot of other modifications. And it used to annoy me when certain people in the field would talk about how, well, you know, 80% of you have a false positive rate of 80% because you can't interpret 100% of your spectra. And so have you searched every single possibility? No. Well, then how do you know that's your false positive rate's that high?
So I have a question. So how important do you think licensing Sequest to Thermo was in the uptake of the program?
In hindsight, I probably would do something different. Thermo, my expectations were not met. My expectation was that, one, Thermo would invest in the software. Two, they would make it widely available. In fact, the license said that they could not deny the sale of it to non-Thermo users. And basically, we had to port the software over to a PC for them because they just weren't investing in it. They just wanted to have it so they could say that they had it.
So did they shut you down in some ways?
From what?
From being able to make it more publicly available?
Yeah. I couldn't give it out anymore. And in fact, they didn't go after infringers anyway. And so the only person who was being held back was me.
John, all coins have two sides, so let me turn the other side of the coin. The development of using the database searches led to the fact that people are no longer looking at these mass spectra, and sometimes they don't even look at the LC-MS runs. So they just get the results of the search, and sometimes you get more protein, sometimes less. What they don't know is whether there is anything else in their LC-MS runs that they have not searched for. So this dark proteome that Bogdan, it's not just unidentified peaks. These are peptides, definitely peptides. You see ammonium ions, you see B2 ions, you see lysine or arginine molecular peaks. So people cannot answer the simple question: what percentage of peptide ion current that you accounted for? Is it 3%? Is it 50%? Is it 70%? And so, can you explain this phenomena that, or comment on this phenomena that people refuse to look behind the curtain, so to say, or they just search under the pole, light pole, and they don't want to look in the darkness, so to say?
When you buy a kit to do molecular biology, do you know what's in the kit?
Sometimes.
I always check for the SDS. in
Well,
that
sometimes they
case.
won't tell you what's in there.
That is the problem.
Yeah, so interesting story. Pretty early on, we're doing DDA, we're doing database searching, triple quadruple mass spectrometer, and Ashley McCormick does a run, searches it, comes back with nothing. And she says, something wrong with the computer program. I said, have you checked the tune on your mass spectrometer? Have you looked at your tandem mass spectra? No. The instrument was way out of tune. So yeah, you're right. I mean, people no longer look at those things, and that I think is a, is that a feature or a bug? I don't know. People really should understand the basis of what they're doing.
Well, that question is actually a good segue to our next speaker, unless you have a burning question for John.
No, a quick question about the AI. I was amazed to see that idea of AI, pop up that early in the people's head. What do you think was current state of AI that, try to help people to find new drugs, to find all these solutions? Is there any artificial intelligence type of program that use our, very sophisticated neural nets and a lot of powerful computers could actually brings us an extra help to do de novo if everything comes out of the
I think you'll
mass-spec?
hear about that next.
Great.
But
Thank
one of the things that I had Jimmy Ng do, mid-90s, was to write a neural net to evaluate the output of the search program. We never published it. And it worked. You either got a 1 or a 0, but it worked. But we never published it. We eventually dropped it, and we started doing other things. But yeah.
I think that's the next step.
Yeah, I had another guy working on neural nets to look at spectra, and Qualcomm hired him, so that ended. But yeah, there will be AI solutions, machine learning solutions. That's one of the things that Bill Noble does very well.
Okay. Well, good luck. I'm looking forward to it.
Well, so to follow up on what John was saying, that not all ions and not all spectra correspond to peptides. I have certainly seen direct evidence for that in some of our single-cell runs, especially when sample prep fails. And in between examining the spectrum by hand and looking just at the sequence identification, there's also the level of looking at all sorts of informative features of the spectrum. And this is one of the things that I'm very grateful to MaxQuant, where we can export aspects such as the how close to the apex of an elution peak we were able to sample the peptide and all sorts of other informative features of the peptide spectrum matches.